So thank you very much for the opportunity to talk at this year's York Talks. My name is David Kent, and I'm based at the York Biomedical Research Institute at the University of York. And our lab really focuses its attention on blood stem cell biology. We're interested in how blood stem cells are regulated and how subversion of blood stem cells drives cancers in people. Now, blood production is not a simple process. In fact, we are producing and destroying every day trillions and trillions of blood cells. And this production and destruction is happening in a very balanced way, where if you came back tomorrow and counted the blood cells in your body, you would have approximately the exact same number of blood cells today as you did yesterday, as you will next week. And this enormous production and destruction is ultimately regulated by the blood stem cell compartment, which we focus our attention on. So as you can imagine, with this enormous production and destruction of blood cells at the scale of trillions, there's a lot of things that could potentially go wrong. And really what I'm about to tell you about today is a recent technology where we're able to discover the sort of intricacies of individual blood cells amongst those trillions to really try and expose what's happening on a cell by cell level and to try and understand what's happening as those cells are corrupted to drive cancers. And we can reconstruct now the relationships between those cells, how one blood cell is related to another cell. And really what we're trying to do is understand this complete family of blood cells. And these errors that happen, these mistakes that happen as blood cells are produced and destroyed in the trillions, are the sort of dirty little secrets that hang about in the blood cell family tree. And what I mean by that is these secrets may stay hidden for decades, and they may never get discovered at all. And now I'll draw a short analogy. If you can imagine, if we wanted to discover the secrets in our own family trees, we might try to bring together a collection of these people and sort of study them in the same space. And what better opportunity to do that than at major events such as weddings, where instead of the 14-year-old cousin Jimmy that you might remember, Cousin Jimmy has turned out to have a penchant for Cabernet Sauvignon and a little bit of spontaneous air guitar. Now, Cousin Jimmy has disrupted this individual wedding as a surprise event. He's disrupted this process of family gathering, and you didn't expect it to happen. Now, if you can imagine a normal family tree with normal individuals and an occasional Cousin Jimmy disrupting things, that's probably quite tolerable. But if I can ask you to imagine that this family tree is enormous, it's hundreds of thousands, millions, billions, even trillions of people big. Just imagine the disruption that hundreds and hundreds of Cousin Jimmys could cause, anything from financial catastrophes all the way to some pretty severe poor life choices that have repercussions for the entire family. Well, this is sort of what we're exactly doing in blood cells. And instead of studying a family tree of people, we're studying a family tree of blood cells. And the reason, the ability to be able to do this, the reason we can do this now is because of the way that cells grow over the course of time. Ultimately, we all start from one single cell with one single genome, this sort of master genome that's formed when sperm and egg come together. Now that's a fascinating process in and of itself, but you can imagine that that one cell has to grow into a person. And that process requires thousands and millions and trillions of cell divisions to accomplish itself. And if we simplify this to a great degree, you can imagine that single starting master cell dividing into two cells. And one cell goes off in one direction, the other in the other direction, and they can accumulate different mutations in the DNA. And so we all start with 3 billion letters of genetic code. And each of those 3 billion letters can be changed with every cell division, with every chemical insult, with everything that they're exposed to over the course of their life. And the vast, vast, vast majority of these changes don't do anything to the function of the cell. And so actually what that allows us to do is to use these individual changes as a barcode for tracking cells, because it marks the cell, and this mark is inherited over the course of cell division and growth. And so you can imagine at this 2-cell stage, the cell's going to a 4-cell stage where new mutations are introduced. New trackable mutations are now present in each of the cells that are created from the parents and ultimately the grandparents. Now, of course, our bodies are much more complicated than these 7 cells I've described to you here. And I want you to imagine that there are trillions of cells with trillions of different yet highly similar genomes that are all derived from this one master genome, but are all individually unique and therefore trackable, and probably most importantly, reconstructible into a family tree. And this is exactly the sort of approach that we're now able to use in the laboratory setting, where we can map from all of the cells that we can isolate from a person today and their differences, and understand that they have individually different genomes, and we can relate them to each other and retrospectively reconstruct a family tree of cell production all the way back to conception, or that initial union of sperm and egg. Now, I'll show you a single red line here, and this single red line represents a single genome. And in the year 1999, when I started my training in Canada, this single red line had just been produced for the first time. The first human genome was actually constructed, and it costed hundreds of millions of pounds. If we tried to do the experiment that would be represented here, the sort of 100 to 150 total genomes, we would actually be spending about 7 billion pounds in the year 2000. So something that's not really achievable for a single research lab to investigate. If we fast forward over to when I relocated to the United Kingdom, in those 10 years after that training happens, this has become remarkably cheaper, and now would only cost you 150 million pounds to be able to create this experiment of 100 genomes. And finally, if we fast forward to today, this actually becomes really affordable because of the remarkable and incredible reduction in the scale of experiments able to be done by the remarkable and incredible reduction of cost in creating these genomic libraries. And this technological advance has now made it possible to create single human genomes for 200 to 500 pounds a piece. And this makes experiments like this one that I'm about to describe to you completely possible. And the experiment that we set out to do, and we initiated this 5 years ago, was to take a single set of blood stem cells from an individual who was 60 years old and perfectly healthy. And we took a sample from the bone marrow, which is where all of the blood stem cells live, and we isolated single blood stem cells from that person, and we grew them up to be able to make enough DNA to do a whole genome sequencing library. We did this for more than 100 cells, and we were able to take those 100 cells and use the information in their genomes to reconstruct the entire family tree of blood cell production. So we're able to build these blood cell family trees. And this is what it looks like. And you can again imagine from conception all the way down to 6 years of age, this sort of progression through mutational time or molecular time, where we're tracking the numbers and specific locations of individual mutations in the DNA in each of these 100 or so cells. And what you can see with the individual branching points along this family tree is where stem cells have expanded to make more stem cells and more progeny from their collective population. And what that really means is this particular clone, as we call it, is growing out relative to other clones. But this is a perfectly normal individual here with a normal distribution of cells producing blood over the course of their life. Now, this technology really allows us to, for the first time, count the number of stem cells present in an adult human. And it turns out this number is around 50,000. It allows us, most importantly, to be able to track these stem cells over time. So we can go in a year later, 5 years later, 10 years later, and ask which stem cell clones or which stem cell progeny are actually expanding over time versus getting eliminated. How are they competing over the course of a life of an individual human? And it gives us a complete understanding of the genetic changes in each individual branch of the family tree. So, of course, we're not interested in studying only normal patients, but we're perhaps most interested in studying where this process goes wrong in people. And the diseases that we study in this case are early stage blood cell cancers. And in particular, the disease that we're focused on is Shwachman-Diamond syndrome, which is a rare inherited childhood disease where a single genetic mutation results in skeletal defects and poor growth, and for the blood system results in bone marrow failure. And this bone marrow failure is being driven by a reduction in the number of stem cells, a reduction in overall blood cell production, placing an enormous pressure on the system to evolve and mutate to grow new cells faster. And the price of that excess growth is a remarkably high predisposition to diseases such as leukemia, where in 30% of cases, these children will develop a leukemia. So we applied this technique of building blood cell family trees to try and understand the process of cancer evolution in these people. And I'll remind you of the way that we do this. We take single blood stem cells from a bone marrow sample, and we expand these blood stem cells outside the body to create enough material to then whole genome sequence them and build a family tree. The family tree in this particular patient with Shwachman-Diamond syndrome looks like this. And there are two things that I'll draw your attention to, which are quite exciting from a cancer progression point of view. The first is this rather large branch of cells over here that all have a single common ancestor, but are expanding quite a lot compared to the rest of the blood cells that we've sequenced. And this represents potentially a leukemic clone that will be growing out in this patient, although we're yet to find out why it's growing out or how it's growing out and what speed it's growing out, which is the focus of our future studies. The second thing I'll draw your attention to, marked in red here, are the number of cells that actually bear mutations in key cancer driver genes. So these are genes that we know cause cancer. And in many, many different cells, these mutations are present. Now, the vast majority of blood cancers are actually caused by one single cell growing out and causing the cancer. But in these patients, we can very clearly see that there are multiple possible cancer-causing cells here, and these mutations have existed from a very, very early stage in life. And so they've existed as early as sort of the first few years in life. And that's represented by the early branching points that we can see in this family tree. And this is quite fascinating because it means in cancer patients, we don't just have one cell that goes wrong, but we have many potential cells from which one of those goes wrong and drives the cancer. And so the two main things here are that we have multiple independent mutations for known cancer-related genes, and then we can also see these large expansions that outcompete the other cells. And this is a process that we're really investigating right now to try and identify what's driving the excess growth. So where do we go from here with this new technology? Well, very specifically in cancers, we know that we can assess now stem cell competition over time. We can identify the relative leukemic or blood cancer causing potential in each of these individual cells. So how likely it is to drive a leukemia in a particular patient, and how rapidly it's doing it over time in sequential samples. Because we have the whole genome sequenced, we can actually also start to identify new targets where specific mutations might associate across large patient cohorts with having either progression to leukemia or protection against leukemia. And we can start to design novel therapeutics to be able to address those very specific leukemia targets. And this technology isn't limited to what we can do in cancers. We're actually able to use it in many other settings of blood stem cell biology. And the Bill and Melinda Gates Foundation has recently funded us to work with collaborators at Boston Children's Hospital, Great Ormond Street Hospital, to try and apply this ability to count blood stem cells in a sickle cell anemia gene therapy trial. And the case here is that we don't actually know in these gene therapy trials how many stem cells are driving the sort of rescue of the patient's defect. How many stem cells do we need to target to be able to recreate the blood system in a healthy way? And this is what this technology will finally allow us to do. And with that, I'll close, giving you this sort of sense of what we can do now that we can discover the sort of hidden secrets within family trees of blood, the sort of hidden secrets that might cause cancers, the hidden secrets that might actually increase our blood cell fitness over time. And I'd be remiss if I didn't thank the enormous collection of collaborators that make this sort of work possible. In particular, on the Shwachman-Diamond syndrome patient sample sets, Professor Alan Warren is a world expert that we work with at the University of Cambridge. And of course, anything that involves DNA sequencing in the United Kingdom almost certainly flows through the Sanger Institute, which is a very big genome sequencing institute that was actually responsible for one of the first human genomes ever produced. And that set of people, in particular, Professor Peter Campbell and his team at the Cancer Genome Project, are essential to these types of operations where we're going in and computationally assessing the blood stem cells of people. Our lab is doing all of the cell biology, and we're really focused in doing that at the York Biomedical Research Institute. And we've got excellent funding partners and key collaborators across the world. And we really want to expand this work over the course of time. And we're excited to be able to share it with you today. And with that, I'll take any questions. Thank you.